Our group project, Module 4, Mosaic Copy Number Variation in Human Neurons. DNA replication is the process by which a molecule of DNA is duplicated. When a cell divides, it must first duplicate its genome so that each daughter cell winds up with a complete set of chromosomes. As we learned from the class, DNA replication is semi-conservative. Each strand of the original molecule serves as the template for the synthesis of a complementary strand. When the process starts, a portion of the double helix is unwound by a helicase, forming a replication bubble and two replication forks. After RNA primers are added, molecules of DNA polymerase bind to both strands of the DNA and begin moving along the strand in the 3 to 5 inches direction, using them as templates for assembling two new strands of nucleotides from 5 to 3 direction. The two double-strand daughter DNA molecules should be identical. However, sometimes a change in the sequence of bases occurs due to copying error. Occasionally, DNA polymerase mistakenly inserts the wrong base as it synthesizes a new strand of DNA, which results in a single base change called a point mutation. There are 4 types of point mutations: silent mutation, missense mutation, nonsense mutation, and frame shift mutation. Duplication error also causes copy number variation in genes. The gene copy number is the number of copies of a particular gene in the genotype of an individual. Genes were almost always present in 2 copies in a genome. However, recent discoveries have revealed that large segments of DNA, ranging in size from thousands to millions of DNA bases, can vary in copy number. The researchers performed 2 experiments to confirm that low-level aneuploidy and CMVs occur in single fibroblasts. In the first experiment, single-cell clones were obtained by limiting dilution, and they were expanded to about 20 sister cells over 7 days. As can be seen from the figure, CMVs are observed in one clone. Chromosome misegregation was observed as a gain of chromosome 2 in one cell and loss of electron in a sister cell. In the second experiment, the researchers sought to determine whether mosaic CMVs were also present in FCTX neurons from post-mortem human brains. And the result demonstrate that somatic CMVs are a common feature of neuronal genomes and suggest that relative abundance of different CMV classes may vary among individuals. Single-cell genome analysis is inherently challenging because all existing approaches require amplifying the genome before measurement. But still, the CMVs researchers observed are valid for several reasons. First, the methods used were previously validated. Second, the amount of CMVs reported are orders of magnitude larger than the amplification generated, which means the larger scale deviation in copy number can only be explained by CMVs. There are also several more evidence showing that the conclusion the researchers drew cannot be stemmed from technical factors. They are most likely valid. Neuronal genomes exhibit elevated levels of aneuploidy and retrotransposition relative to other cell types. This finding inspired McConnell's research. The research in mosaic DNA copy number variation started with the speculation that somatic genome variation may contribute to functional diversity in the human brain. 2 methods have been developed to map large-scale CMVs in single cells: microarray analysis of multiple displacement amplification, MDA products, and single-cell sequencing, and 2 human neuron sources: human-induced pluripotent stem cells, HIPSC, human postmortem frontal cortex neurons were used. From the methods described above, two phenomena were observed. Figure 1a and c represents subchromosomal deletion, while Figure 2 demonstrates subchromosomal duplication. The researchers examined neurons from three HIPSC lines, referred to as C, D, and E, that were generated from three different individuals as neurotypic controls for a HIPSC-based disease model. Analysis of bulk DNA from C and D line donor fibroblasts or HIPSC-derived neural progenitor cells (NPCS) revealed no chronogenomic aberration. Of 40 single neurons analyzed, 27 had copy number profiles consistent with bulk DNA, but 13 had unique genomes, 7 whole chromosome gains, 4 whole chromosome losses, and 12 subchromosomal CNVs in 13 HIPSC-derived neurons were identified. Each CNV was identified in merely one neuron, which suggests that the CNVs are not early clonal events but rather are unique to single cells or distinct lineages. The CNVs detected in C and D line HIPSC-derived neurons were distinct from those seen in either C or D line fibroblasts or NPCS. Of 29 fibroblasts, 6 had single CNVs, range 5.2 to 27.7 MB, and 1 was aneuploidy. Among 19 HIPSC-derived NPCS, only 6 duplications were observed. Technical replicates of 5 fibroblasts and 3 HIPSC-derived neurons shown high concordance, and principal component analysis also showed that replicates from each individual neuron clustered distinctly from both the fibroblasts and the other 2 neurons. Comparison of CNVs in the 3 cell types showed that neurons have significantly larger CNVs than fibroblasts. These results show that deletions only in HIPSC-derived neurons and not in HIPSC-derived NPCS.
The importance of this experiment is that it extends the observation of somatic mosaicism to the single-cell level, proves that mosaic CNV is abundant in human neurons, and contributes to the testing of the hypothesis about the effect of somatic genome diversification on neuronal function. This is the citation. Thanks for watching.